Hi everyone, myself Ganesh. I am here to explain about the G2 phase of cell cycle. G2 phase of cell cycle is present in the interphase. It is also called as second gap phase. The G2 phase, also known as the second gap phase, is a stage of cell cycle that occurs after DNA replication has taken place in the S phase and before the onset of mitotic M phase. During this phase, the cell undergoes further growth and prepares for cell division. Here are some key facts about the G2 phase. DNA damage checkpoint. During the G2 phase, the cell checks for DNA damage that may have occurred during DNA replication in the S phase. If any damage is detected, the cell will attempt to repair it before proceeding to the M phase. Protein synthesis. The G2 phase is a period of intense protein synthesis during which the cell synthesizes the proteins needed for cell division. Centrosome duplication. Centrosomes, which are important for the organizing the microtubules that pull the chromosomes apart during cell division, duplicate during the G2 phase. Mitotic spindle formation. The G2 phase also marks the beginning of mitotic spindle formation as the microtubules begin to organize into a spindle-shaped structure that will separate the chromosome during mitosis. Checkpoint for mitotic entry. The G2M checkpoint is a crucial control point that ensures that the cell has completed all necessary preparations before entering into the M phase. If the cell fails to pass the checkpoint, it will delay entry into M phase until it is ready. Overall, the G2 phase is a critical period of cell cycle that ensures that faithful transmission of genetic material to daughter cells during cell division. Before the G2 phase. Before the G2 phase comes the synthesis or the S phase. One of two major steps for the cell cycle is prepared here. The genetic material embedded in the DNA is replicated. The cyclins and cyclin dependent kinases CDKs are responsible for the transition for the S phase to G2 phase. After the G2 phase. After the G2 phase, mitosis promoting factor or MPF is secreted, enabling the M phase to start. As the M phase starts to neutralize, the nucleus divides to form daughter nuclei around it. The cytokinesis supplies the protoplasm for the two daughter cells. The mitosis is divided into 5 subphases, namely prophase, prometaphase, metaphase, anaphase, and telophase. For the G2 phase and the beginning of the M phase, before sister chromatins separate, foreign DNAs are found in the cells. G2 phase consists of a cell cycle follows the synthesis where genes are multiplied and DNA is found as chromatins to form separate chromosomes again in the M phase. It is a part of the preparation process that a cell undergoes before splitting into two, and it is intermediate phase between the S phase where genetic materials multiply and the M phase where daughter nuclei are born. G2M checkpoint. There happens to be various proteins accumulated at the broken points of the DNA strands. Where these are visible, the cell is not permitted to replicate. Another concern evaluated in the G2M checkpoint is whether all DNA strands are doubled. This phase also checks if there are enough organelles in the right order. A cell growth can falter under stress. Using this notion, UV light is used to abort the functions of the cancer cells. Conclusion. The G2 phase is the second gap phase in the growing and preparatory period of a cell's life. It helps and checks cell activities before cell division in the mitosis phase can begin. The growth period of the cell cycle or interphase is spread over 3 phases, G1, S and G2. G2 is the last or second growth period after which mitosis or cell division happens. After that, the cell enters the M phase, where it is engenders two cells by dividing itself. The new cells repeat the same behavior provided they pass all intrinsic barriers set by the cell to ensure only the fittest are allowed to multiply. Thank you everyone.